Hello, my name is Simply True, and today is Sunday, May 30th, 2021, and today I will be doing a video of lab number 5, Bacterial Morphology for about 25 microbiology. So in the first exercise, I had to go ahead and take a swab of my inner cheek and then place it on these two slides, one for the direct stain and one for the negative stain. So with the direct stain, I went ahead and put the specimen onto the slide, and then afterwards I had to heat fix it. So I had to put it over the flame for about 5 times to go ahead and heat fix the bacteria onto the slide, and then afterwards I went ahead and put a drop of the crystal violet dye, which is a basic dye, and then afterwards rinse it off, and then I went ahead and took a picture of it and uploaded onto the lab. For the second part, same thing, I went ahead and swabbed my cheek and then put it on the slide. And for this one, since it's a negative stain, I do not need to heat fix it, and then all I had to do was put a drop of the Congo red, which is the acidic dye, and after that washed it off, and then if you look at it in an angle, you would be able to faintly see the residue. And the whole point of these two stains is that the direct stain, it needs heat fixing, because when the crystal violet, which is a basic stain, is added to the slide, it actually stains the bacteria itself. And then for the negative stain, it does not need to be heat fixed, because the Congo red is an acidic dye, and then when it's added onto the slide, the whole point is to actually stain the background, but leave the bacteria alone. And for this part, it was a bit successful. If you look really closely for the direct stain, you can see like a light purple dot in the middle, which is the bacteria itself that's been dyed. And then for the negative stain, it was successful also, because if you look at it closely, you can see the orangish residue of the stain on the background. Then for exercise number 2, I have to go ahead and do the swab of the E. coli, S. epidermidis, and also S. cerevisiae, or yeast. So for this part, I had to go ahead and take the cultures from the other lab in the broth, and then I had to go ahead and use the inoculated tube, sterilize it, dip it into the broth of each one, and then go ahead and put it to the according slide. And then for each of those, I had to go ahead and did I heat fix it? I did heat fix it for each one, so I had to pass the slide over the flame for about 5 times, let it cool, and then I did the gram steaming technique, which follows 4 steps. So after the specimen was heat fixed on each of the slides, I had to go ahead and let it cool. And then after that, the first stain that was added was the crystal violet stain. I let it sit for about 60 seconds, and then I washed it off with water, or rinsed it off with water. After the crystal violet dye, it leaves it kind of purple, and then once it's washed off, I went ahead and added the iodine, which leaves it kind of like a brownish color. Left that on for 60 seconds, and then after that, also rinsed it off. After the iodine, I had to add like a decolorizer, which is a type of alcohol, or ethanol. I left that on for about 30 seconds, and then it's kind of like a clearish color. Had to rinse that off, and then finally I added the saffron in, which is like a goldish dye. Left that one on for 30 seconds, and then I rinsed that off. And then afterwards, I let each of the slides sit to dry completely. And then after that, I went ahead and took a picture and uploaded it to the labs. But for the E. coli one, it wasn't really successful. Same with the S. cerevisiae, wasn't really successful. It's pretty much clear, but the S. epidermidis, for some reason, you could actually, if you look closely, you could kind of see like the orangish residue that's left behind. And the reason why I think it wasn't successful was because I was using the cultures from the previous labs, and during the previous labs, when I was trying to incubate the culture in the broth, wasn't actually successful in seeing any microbial growth for the last lab experiment. So since I wasn't able to see any microbial growth in the last lab experience, I'm assuming that's why I wasn't able to get a good culture for these slides, and therefore I wasn't able to see it after even after the gram staining technique. And then also, this experiment is applicable in real life, because if you're working in like a research facility, you would need to know and be familiar with the gram staining technique, which includes, once again, 4 steps. So first you would have to heat fix it, then you would add the crystal violet dye, which is a basic dye, turns it purple, and then after the crystal violet dye is added and rinsed off, you would add the iodine, which makes it a brownish color. After that, you would rinse that off, and then you would add the decolorizer, which is usually an alcohol, and it leaves it looking clearish. After that is rinsed off, you would add the saffronin finally, which is also like a golden color, and you would rinse that off, and then afterward you would be left with the specimen on a slide. And from this experiment, I learned that there is 2 different types of bacteria, gram-negative and gram-positive. The main difference between gram-negative and gram-positive bacteria is that in gram-negative bacteria, it has a thin peptidoglycan layer, and it also has a an outer layer to it. But for the gram-positive bacteria, it actually has a thick peptidoglycan layer or membrane, but it doesn't have an outer layer. And I also learned that the bacteria that's more easily treatable with antibiotics is actually the gram-positive bacteria, because it has a thicker layer, and that thick layer is able to absorb the antibiotics better. So that's why it's easier to treat gram-positive bacteria with antibiotics. And then for future reference, in order to conduct a successful experiment, I would actually have to be able to have been successful with the other lab experiment, which was culturing the E. coli, S. epidermidis, and S. cerevisiae properly in the broth medium. I would have to be sure that I was able to see microbial growth, because seeing microbial growth means that I actually have a good amount of bacteria in the broth. And if I had that, I would have been able to have good samples for this lab.